morning, viewer. Welcome to our Glory Globe TV has some breaking news. Yeah, in many cells of the human body, hair-like protrusions are known as cilia act as antennae, allowing cells to receive signals from their environment and other cells. As cells grow and divide, each cilia must first form on a cell body, then disassemble or break down by shedding or shortening before the cell divides. Many genes involved in cilia formations have been previously studied, but mutations in these genes are known to cause a host of pediatric disorders or ciliopathies that impact the formation of bodily systems like the skeleton, heart, and brain. However, presently scientists have known little about which genes play a role in allowing cilia disassembly to occur, as well as how defects in the process impact the body. In a new study, a Yala research team has identified a series of genes that make up a pathway responsible for the disassemble of primary cilia, that is called a type of single immobile cilia found in some cells, and when defective may be linked to a neurological disorder called focal cortical dysplasia. The results are published in Science Advances. Our goal was to use new genomic technologies to systematically approach the questions of cilia disassembly, said David Breslow, an associate professor of molecular, cellular, and developmental biology in Yale's Faculty of Arts and Science, and the study's corresponding author too. We uncovered fundamental aspects of how cells work that had not been well understood, as well as a potential new connections to a disease that could help us understand its causes and therapeutic strategy. Studying cilia disassembly could lead to better therapies for neurologic disease as well as other conditions including cancer, to which the abnormal breakdown of cilia might contribute. Previously, Breslow said members of his lab began using CRISPR clustered regularly interspaced short palindromic repeats, a gene editing technology to identify the genes needed for cells to build primary cilia. To do so, they knocked out, that means inhibited, the function of every gene in the genome, a technique that allowed them to link certain genes to diseases associated with faulty cilia formation. Amazing, sir. Beyond this exclusive report, Glory Globe to Delhi, Bharat.